Hey there, Jeremy Martin here, and we're going to talk today about ion channels and some neural communication, which ultimately is signal transduction. Click. Cool. So starting with a little bit of the purpose here. Why is signal transduction so important through neurons? Well, the overall purpose of this electrical signaling is for communication between the external environment and one's internal environment, as well as communication within one's internal environment. The sensory neurons get information about what's going on inside and outside of the body and bring that information to the central nervous system so it can be processed, I should say. For instance, if you picked up a hot coal, sensory neurons with endings in your fingertips would convey information that it was really hot. Interneurons, which are found only in that central nervous system, connect to another neuron, like the sensory neurons, and they receive information, again, either from the sensory neurons or other interneurons, and transmit that information to motor neurons or other interneurons. Back to that hot coal, the signal from those sensory neurons in your fingertips would travel to the interneurons in your spinal cord, right? And some of these interneurons would signal the motor neurons in your muscles, making you release the coal, while others would transmit the signal up the spinal cord to the neurons in your brain, where it would be perceived as pain. Motor neurons get information, again, from other neurons and convey those commands to your muscles, organs, or glands. So when you release that hot coal, that's the motor neurons activating. Dendrites, get that off of there. So a little bit about the form of these neurons, starting with the dendrites. They're often viewed as sort of the top of the nerve cell, depicted as a heavily branched region with pointed ends. This side receives and processes excitatory or inhibitory information in the form of neurotransmitters from other nerve cells. This determines if an action potential will be relayed down the axon to the axon terminal on the other end. The soma is also called the cell body, and this contains the nucleus and other organelles for basic cell function and joins the axon at an area we call the axon hillock. The axon is that elongated, skinnier region, oftentimes covered by a myelin sheath for insulation and creates more rapid signal transduction. The axon ends at the axon terminal, which is slightly branched with a bulbous ending compared to the dendrites, which again were heavily branched with pointed endings. The terminals with the target are going to interact with the targeted tissue or another neuron to continue transmitting that signal down the line. Within the cell membrane of the axon terminal are neurotransmitter reuptake pumps to remove the chemicals from the synapse and bring transduction to a halt. Synaptic vesicles are membrane-bound sacs of neurotransmitters packaged for release into that synapse when an action potential is sent through the axon. So with regards to these receptors, a synapse is a connection between two excitable nerve cells, where a neurotransmitter is released from the presynaptic cell and diffuses across the synapse and binds to a receptor on the postsynaptic cell membrane. This is going to convert that electrical signal from the action potential in the presynaptic nerve to a chemical signal in the form of neurotransmitters in the synapse, which is going to generate a new electrical signal in that postsynaptic cell. There can be hundreds to thousands of synapses between the axon terminal of one nerve and the dendrites of the following postsynaptic nerves. On the postsynaptic nerve cell, there are typically two types of general sort of classes of receptors. These Metabotropic receptors are those which generate and release secondary messengers by changing conformation. And these G protein coupled receptors, which are relatively slow in the realm of signal transduction, since they work through certain chains of secondary messenger molecules. These are notably the most diverse protein receptors in eukaryotic cells. And some examples include the muscarinic acetylcholine receptors, all types of neuropeptide receptors, and most biogenic amine receptors. Ionotropic receptors are the other type, the other sort of class, which are those which respond to a bound ligand, so essentially a neurotransmitter, right? These are also called ligand-gated ion channels, which operate much more quickly than the metabotropic because their transduction does not include those secondary messengers, and their effect takes place immediately as ions move in or out of the cell. These could be exemplified by the nicotinic acetylcholine receptors, most of the amino acid receptors, and some purinergic receptors as well. Voltage-gated ion channels also work very quickly, just like the ionotropic receptors, but they open and close depending on the membrane's potential, that electrical potential. The three primary types of voltage ion-gated channels are the sodium, potassium, and calcium-specific channels. Some different types of neurotransmitters. I'll start with saying that depending on the type of neurotransmitter and the receptor combination, the signal will be considered excitatory or inhibitory. Some compounds fall into one of the two categories, but others can have varying effects on the cell depending on the receptor it's bound to. Cells are typically specialized in that they only make and release one type of neurotransmitter. This is called Dale's law. A cell types, all cell types, I should say, are often classified based on the neurotransmitter they make. So a cell that makes and releases dopamine is known as dopaminergic, a dopaminergic cell. One that makes and uses acetylcholine is cholinergic, glutamate is glutamatergic, and etc. Different classes of neurons can make different types of neurotransmitters, and every neurotransmitter has its own specific type of receptor. So, for example, cells that release the acetylcholine activate at least two different types of acetylcholine receptors, which have very different effects on the postsynaptic cell depending on which one is there and bound. So, for example, the nicotinic acetylcholine receptors are ion channels which open when bound to acetylcholine, and they're going to let positive ions into the cell for depolarization. Typically, these are sodium ions. This type of synapse is the one found on the neuromuscular junction, the place where neurons are going to be contacting with muscle cells. In contrast, the muscarinic acetylcholine receptors, when they're bound with acetylcholine, they will activate a biochemical reaction that leads to the opening of an ion channel, which will let positive charges out of the cell, in other words, causing hyperpolarization. So the action of this neurotransmitter depends on the receptor. They have perfectly opposite effects on that postsynaptic nerve. And as I previously stated, some secondary messengers are intracellular ligands used for signal transduction by the G protein coupled receptors. When these receptors are bound by a neurotransmitter, they're going to release a secondary messenger or directly interact with intracellular enzymes or other membrane bound channels to pass this signal along. So with regards to excitation or inhibition, excitation, I should say, or inhibition, right? These neurotransmitters can really be thought of as extracellular ligands. They're going to stimulate ion channels to either open or close. They can be excitatory, where they influence the cell to transduce a signal, or inhibitory, where they prevent that signal from being transferred. Some of the neurotransmitters, some excitatory neurotransmitters include adrenaline, which is primarily a hormone released by the adrenal gland. Some neurons may secrete it as a neurotransmitter, though. It's going to increase heart rate, increase blood flow, leads to a physical boost and sort of heightened awareness in an organism. It's produced during stressful or exciting situations a lot of the time. Glutamate would be another one of these excitatory options, which is the most common neurotransmitter in the brain, actually. It's responsible for regulating development and creation of new nerve pathways, hence why it tends to be involved in learning and memory. Some other excitatory neurotransmitters could be aspartate, nitric oxide, or ATP. On the inhibitory side, those neurotransmitters could include GABA, which is typically found in the central nervous system, where high levels are associated with improved focus, whereas low levels tend to cause anxiety. Or serotonin, another example, which contributes to feelings of like well-being and happiness. And these are affected by exercise. It's going to play a role in sleep, as well as throughout the digestive system for regulation. Glycine can also be an example of an inhibitory neurotransmitter. Some of those that sort of depend on the type of receptor they're bound to are going to include noradrenaline, which, in contrast to adrenaline, noradrenaline is predominantly a neurotransmitter that's released as a hormone occasionally, mostly neurotransmitter. It contracts blood vessels and increases blood flow. It's going to be associated with improved attention and the speed at which a response can then occur. So more of it, quicker the response. Dopamine is also sort of dependent. It's primarily responsible for feelings of pleasure. It's also involved in movement and motivation for humans. People tend to repeat behaviors that lead to dopamine release. So dopamine is essentially associated to addictions. And when abnormal dopamine secretion occurs, it's common in sort of movement disorders like Parkinson's, for example. Acetylcholine is another sort of dependent example here. It's involved in thought, learning, and memory within the brain. It also activates muscle contraction in the body and is associated with attention as well. Let's talk about how this sort of movement of the signal actually occurs through the voltage and membrane potential. This membrane potential is the difference between the intracellular and the extracellular fluid. A neuron at resting membrane potential, is negatively charged down to about negative 70 millivolts. This depends on the type and the location of neuron, though. If a membrane were equally permeable to all ions, each type of ion would flow freely across the membrane and the system would reach equilibrium. That's no good, right? Because ions cannot simply cross this membrane, though, the different concentrations of sodium, potassium, and chloride ions inside and outside the cell allows for this signal transduction. The difference in the number of positively charged potassium ions inside and outside of the cell pretty much dominates this whole membrane potential game. When the membrane is at rest, the negative charge within the cell is created by the actions of the sodium-potassium ATPase pump, as seen in the image here. The energy for the movement against the electromagnetic gradient comes from ATP hydrolysis. So for every molecule of ATP that's broken down, 3 sodium ions are moved to the outside of the cell and 2 potassium ions are allowed in. Because more positive ions are removed from the intracellular space than are taken in, the inside of the cell remains negatively charged relative to the extracellular fluid. The chloride ions, which are not shown in the image here, the chloride ions tend to accumulate outside of the cell because they're repelled away by some negatively charged proteins that are found throughout the cytoplasm. The direction of spontaneous ion flow across the polarized membrane is dictated by two factors: the concentration gradient and that electromagnetic potential gradient. As resting nerve cells remain around negative 70 millivolts, calcium and sodium ions will spontaneously flow inward. So when channels open, they naturally come in, but potassium and chloride ions are going to naturally flow out when their designated channels are opened. Potassium flows out despite being against its electromagnetic gradient due to the higher intercellular concentration. So more potassium inside forces them out. Chloride, on the other hand, flows outward more predictably due to that electromagnetic gradient sort of pushing it outward. When calcium ions are used in this way, it's mostly in sort of specialized locations such as myocytes. Depolarization is when one of the neurotransmitters that we discussed 2 slides ago binds to the receptor of a ligand-gated ion channel, and an excitatory response is initiated, and the cell is then said to become depolarized. This excitatory response brings the membrane potential up closer to 0 or above 0 millivolts by the opening of these sodium channels. Occasionally, calcium channels are used, like I said. This is going to allow for the designated ion to flow into the neuron against their concentration and their electromagnetic gradient. The cell being considered the postsynaptic cell, I should say. Therefore, we call this change an excitatory postsynaptic potential. If the concentration of the neurotransmitter in the synapse is high enough, or in other words, if there are enough of them binding to the receptor proteins and the potential rises above roughly like negative 50 millivolts, an action potential is then triggered and the electrochemical signal is sent down the axon. Most of the time, the single excitatory postsynaptic potential is not enough to trigger a full action potential because they're basically graded through the soma, which we'll discuss next. These graded potentials are wave-like excitatory postsynaptic. Let me move this down. Postsynaptic. Postsynaptic potentials generated by sensory input to the body. This begins in the dendrites of the neurons. They're considered graded because their depolarization can range from about 1 to 50 millivolts. So it's an increase from that negative 70 by about 1 to 50 millivolts. The difference depends on a few factors, including the number of neurotransmitters in the synapse, as discussed in the previous slide. Could depend on the length of time that the neurotransmitters are in the synapse, and as well as how long the neurotransmitter might be bound to the receptor protein across the synapse. The more neurotransmitters in the synapse, or the longer they're there, or the longer they're bound, the larger the excitatory postsynaptic potential is, and the more likely it is that the neuron will proceed with an action potential. Multiple graded potentials are often required for an action potential to initiate because the strength of the electrochemical signal fades as ions diffuse through the soma toward the axon hillock. We kind of see that in the image there. At the same time, voltage-gated chloride ion channels are going to open to sort of counter this depolarization. So two things working against it, the diffusion as well as those chlorine ion channels. In other words, the firing of an action potential down the axon is dependent on the sum of ions collected at the axon hillock as they passively move through the dendrite. In order for an action potential to occur, there's this all or nothing or all or none phenomenon that kind of needs to happen. In other words, the requirement of enough depolarizing graded potentials to build positive ion concentration compared to the negative ions at the axon hillock. So the comparison between chloride and sodium ions in most cases. Once the excitatory postsynaptic potential reaches about negative 50 millivolts, again, that action potential is initiated and sent down the axon. In the same way, if the graded potential does not sum to negative 50 millivolts, there's just no action potential, and the signal transduction is going to end right there. Action potential can be triggered by two different sort of summations. Temporal summation, where one region of the dendrites receive excitatory interactions in rapid succession. The other version being spatial, where many excitatory potentials occur throughout the neuron's different dendrites. This is essentially how neurons are able to determine whether a signal is worth transduction or not. A single neuron can create a variety of action potentials depending on the type of ion channels open and the location of that neuron. So once an action potential is initiated and signal transduction is sort of continued through this nerve, voltage-gated sodium ion channels open sequentially down the axon to its terminals, as shown in the bottom image. When the membrane potential labeled on the x-axis reaches about negative 50 millivolts, there's a sharp increase up to about positive 30 millivolts, which is going to represent the action potential being triggered. Notice the direct correlation with an increase in sodium ions as they flow rapidly into the cell with the opening of those voltage-gated ion channels. This is going to create a sort of positive feedback loop down the axon where the increase of sodium ions opens channels in front of the action potential. At the same time, potassium ions are constantly sort of leaking out. Again, sort of in front of the action potential, and they're going to try to maintain this balance of concentration between the electromagnetic gradient and overall concentration. But these do not succeed as more sodium ion channels are open, so there's more influx of sodium than there is efflux of potassium. In other words, the depolarization of rapid sodium ion influx is trying to be countered by some hyperpolarization by the efflux of those potassium ions. So when the signal gets through the axon and arrives at the axon terminal, the voltage-gated calcium ion channels are going to open to allow this calcium ion to rush inward. The calcium ions are going to trigger a reaction in this terminal, releasing synaptic vesicles to be moved toward the plasma membrane for release of the whatever particular neurotransmitter is being signaled. The neurotransmitters diffuse across the synapse to bind to receptors on the dendrites of the postsynaptic neuron, and the process repeats itself in the new neuron, assuming that a graded potential is strong enough to trigger the action potential. Once the signal is delivered, excess neurotransmitters in the synaptic cleft either drift away, they're broken down into inactive fragments, or they're reabsorbed by the reuptake pumps of the presynaptic membrane. Clearing the synapse is important. It serves to both provide a clear on and off state between signals, and it's going to regulate the production of neurotransmitters. Because after reuptake, the full synaptic vesicles are going to provide signals to that cell that no additional neurotransmitters are needed to be produced. At this point, hyperpolarization is, which is also considered an inhibitory potential, is going to start to occur. As the membrane potential moves down from that plus 30 millivolts to 0 and then continue back down to about negative 70, This is, again, hyperpolarization. After an action potential, this sudden increase in membrane is going to actually trigger at about 30 millivolts above zero. It's going to trigger the closing of those voltage-gated sodium ion channels. This does not just occur as voltage-gated ion channels are open to let potassium out. I should say this, at the same time, these potassium channels are opening to let them out, but in unison with the voltage-gated chloride channels, they're going to open and let those negative ions in. So to rephrase, the potassium channels open to let that positive, the cations out, and the chloride channels open to let those negative anions in. Again, trying to make this more negative. It's going to help speed up the process of returning the membrane potential to about that negative 70. This primarily occurs in neurons as they counteract the effects of the excitatory postsynaptic potentials. So it's just bringing it back down to resting state. Hyperpolarization is most often initiated by the inhibitory neurotransmitter we mentioned before, GABA, like the image depicts right here. When the type of chemical is bound, GABA is bound, the hyperpolarization brings the membrane potential away from reaching that threshold for action potential as it becomes more negative. In this way, the excitatory and inhibitory postsynaptic potentials can actually combat each other, where the stronger of the two is going to determine how that neuron responds and whether or not signal transduction continues. So between action potentials is this refractory period. It's essentially after an action potential, the neuron struggles to produce another action potential because it's got to transition back to a resting potential from that depolarized state. During this period, hyperpolarization is occurring so that the voltage-gated potassium and the chloride ion channels are all going to be open. This is going to now allow for their natural efflux of the potassium and influx of that chloride I was just kind of talking about. The voltage-gated sodium channels, again, are still all closed up. This time, this time period is going to limit the number of action potentials that a neuron can undergo. So although different neurons can hyperpolarize at different rates, there's almost always a lag time. This sort of depends on the size of the neuron as well as the number of and type of ion channels that it contains. The refractory period is also why an action potential cannot travel in reverse up the axon, since the voltage-gated sodium ion channels are all closed, and we're getting rid of all those positive ions. Here are some of my references, as well as some for the pictures. Take care.